Okay, so we are going to start with the section Other Proteins that Assist DNA Replication. We may have done a little bit of this in class, but it's a good review anyway, depending upon what hour you were in. So we have, remember that the DNA has to untwist, so you have helicase, untwists the double helix, and then breaks the hydrogen bond, separates the template DNA strands at the replication fork. So that's your original strands. And then the single-stranded binding proteins help to stabilize and keep those two sides from re-hydrogen bonding together, so it keeps them apart so that you can build on each one of those. And so then you have topoisomerase, corrects overwinding ahead of the replication fork by breaking, swiveling, and rejoining the DNA strands. And so therefore, after DNA replication, it helps to retwist and so on. Primase synthesizes an RNA. You should be able to fill this blank in. Should be an RNA primer. When I ask questions like that, if you really want to see if you understand it, you should pause the video and see if you can figure it out if I don't give you enough time. Because some people, it may take a minute to kind of pull it out of your head. You may know it, but I may do it too fast. So you can go ahead and pause it. Then blank continuously synthesizes the leading strand and elongates the Okazaki fragment. So what synthesizes the leading and elongates the Okazaki fragments? They add nucleotides. That is DNA polymerase 3. Then blank removes the primer from the 5' end of the leading strand and the Okazaki fragments and replaces the primer with DNA, adding to the 3' end. So what replaces the primer with DNA? That is DNA polymerase 1. And blank joins the 3' end of the DNA that replaces the primer to the rest of the leading strand and also joins the lagging strand. We should know that. What glues them all together? DNA ligase. All right, so this picture here pulls it all together and shows all the different parts. So this part right here, a lot of this is the same as the previous picture that we had talked about. And so you have one primer, it's not showing you here. This is the 5' end of this strand here. And this is the 3' end of the, so that means this is the 3'. So it's moving. This is why that's the leading strand. And it's going to, DNA polymerase 3 continues to add the nucleotides. There would be that one primer here. Here are your Okazaki fragments. And so you had a primer here and DNA polymerase added, new primer, DNA polymerase added, new primer, DNA polymerase added. All right, then coming back, DNA polymerase 1, it's already, there's no primer here anymore. DNA polymerase 1 has already replaced that RNA primer with DNA. And so now it's replacing this one. And so this is all gonna be DNA in just a minute. And then it'll come and replace this primer. And here's the primer, and this DNA polymerase 3 is currently adding nucleotides. And so we have all these Okazaki fragments, and in comes DNA ligase. Once the DNA polymerase replaces the RNA primers, then DNA ligase will hook the two strands together. And so that's what we have here. And then a couple of things that are not labeled that I want to label here. This right here is at the very beginning. This is, what do you think that is? What untwists and unwinds the DNA? It's helicase. And these guys right here, these guys stabilize and keep the DNA separate so that DNA polymerase and primase can do its thing. These are the single strand binding proteins. Good. All right, and so So moving on, so that's how DNA replication occurs. And so, real quickly, this shows you a replication bubble. And so when you're looking at these two sides here, we're looking at inside of this box. But remember earlier we talked about a bubble, and so here's the origin of replication. And so here you have your primer in dark here, and then this is your leading strand here moving towards the replication fork, which means then that you have fragments, Okazaki fragments, in your lagging strand. And so you have a primer here, Okazaki fragment, primer here, building the Okazaki fragment in opposite direction. But then notice here, if we looked at this side now becomes the leading strand. You have one primer and it moves in continuous direction in the 5 to 3' direction. And here you have multiple primers to grow in the 5 to 3' direction in this case here. So it depends upon which way you're looking at it. So this is growing towards the replication fork and this is away. So therefore this is the lagging strand on this side and this is the leading strand on this side. And then DNA replication would occur in both directions. Remember, it's kind of going back to what picture? This picture here. So that DNA replication occurs very quickly and you end up with two daughter molecules much quicker than if you just started at the one end. Okay, so let's look at proofreading and repairing DNA. DNA polymerases proofread newly made DNA, replacing any incorrect nucleotides. What does that mean? If it accidentally matched an A with a C, A's and C's don't go together. A's go with T's. So if you had a T coming out of your template and it mistakenly added a nucleotide with a C here, that's wrong, right? And so therefore you have enzymes that help to fix those mistakes. So in mismatch repair of DNA, repair enzymes correct errors in base pairing. So that's what I meant here, was trying to illustrate there. In the next kind, nucleotide excision repair. Oops, I don't have to write repair, it's already there. Enzymes cut out and replace damaged stretches of DNA. So if we look at this next picture here, this next picture illustrates that. So this is called a thymine dimer. It's two thymines. Notice here what's going on. They're bonded to each other this way. They're supposed to just hydrogen bond this way, and the phosphates and the sugars of the backbone are supposed to be connected, not the actual thymines. And that distorts the shape. So you see this distorted bump here. It distorts the DNA molecule. So you have an enzyme called a nuclease. It cuts the damaged DNA. So this is the nuclease right here, this enzyme. It cuts right here and right here where those arrows are. So before and after where the mistake has occurred, the damaged part. And then now you're missing some nucleotides. So in comes DNA polymerase and fills in the missing nucleotides with the correct nucleotides. So A's and T's go together and C's and G's. So they fill in the missing nucleotides and then they have to be glued together right here and right here once you get at the end. So DNA ligase, we know, seals those and causes those to bind together. And so that's an example of this is the nucleotide excision repair. Enzymes cut out and replace damaged stretches of DNA. Mismatch was what I drew here when there's an error in base pairing, an accident. So we have lots of enzymes that kind of proofread and fix our mistakes so that hopefully at the end of DNA replication we don't have anything wrong. All right? So the imperfect nature of DNA replication and repair can increase variation. Let me explain how that works. All right, so let's say you have a piece of DNA. So here's your DNA. Remember, a gene is a section of DNA. And that gene codes for making a protein. So here's gene A, makes protein A. So then let's say that something comes in and causes a mutation. So what's a thing that's a mutant can cause a mutation? UV light. So let's say UV light induces a mutation. All right, so that's what we have going on. So that means the order of the bases, A's, T's, C's, and G's, there's a mutation there. So after the mutation, what's it going to look like? So let's write after the mutation. So let's draw our DNA again. So here's my original DNA. It's my blue DNA. Now the gene is mutated. So here's your gene here. But now it's a mutated gene. All right, so now if it's mutated, you could have no protein made because it no longer codes for making the same protein because the order of the bases have been changed. That is a bad mutation because whatever protein that was supposed to do, whatever job that protein was supposed to do is no longer being done. Can a mutation ever be good? Absolutely. So sometimes you can have a new protein made. And if that new protein that's made is good, then we have an increase in variation. So this, I'll put here, may be good or bad. Or maybe a little bit of both. Maybe you don't have this protein anymore, so that's a bad thing, but then you have a new protein that does something else that's helpful, so that would be a good thing. So therefore, all genetic variation comes from random mutations and whether or not they're good for survival in the environment or not. And some of that mutation, when it says the imperfect nature of DNA replication can increase variation, I showed you UV light changing the gene. But sometimes if you get a mistake in, let's say, a mismatch or a mistake in DNA replication, that's another source of variation where the gene isn't necessarily mutated, but it's definitely by an outside source, but it's due to DNA replication, and that can have the same effect. And then lastly, we have one last thing that poses a problem, and that is the ends, the very ends of the DNA molecule. So limitation of DNA polymerase creates problems for the linear DNA of eukaryotic chromosomes. We have like linear DNA versus bacteria that have circular DNA. So this problem is for eukaryotic chromosomes like our cells. The usual replication machinery provides no way to complete the 5' ends. So repeated rounds of replication produce shorter DNA molecules. So eukaryotic chromosomal DNA molecules have at their ends nucleotide sequence called telomeres. These are non-coding sections of DNA, and this is at the very end. So in humans, we have the repeat sequence T, T, A, G, G. So this happens over and over and over again, all right? And it doesn't code for making a protein. It's just the sequence of bases at the ends of our chromosomes. So what this does is it does not prevent the shortening of DNA molecules, but they do postpone the erosion of genes near the ends of the DNA molecules. So let's draw this before we do these last little bits so that you understand what's going on here. Let me get a black. All right. I'm going to draw up here since we have some space. So let's say we have our generic DNA molecule. Here's the two sides, A's and T's, C's and G's. This is the 5' end, so that would mean this is the 3', and this would be the 3', and this would be the 5'. So if we just look at, in general, DNA replication, so that's going to untwist and unwind. What enzyme does that? Helicase. So here we have our two sides. So this is the 5', this is the 3', this is the 3', this is the 5'. All right, so they've untwisted and broken the hydrogen bonds here. So then, where's replication going to occur? Well, remember the new strand, where does it start? It has to grow in the 5' to 3' direction. So this is going to be the 5' end and the 3' end of the complementary strand. And so therefore you have to have a primer. So primase will add your primer, so that's your RNA primer. And then DNA polymerase can add and grow in the 5' to 3' direction. So that's what we have here. On this side, we have this is going to be the 5' end, and this is the 3' end. So we're going to have a primer here, and then grow this way. All right, and one is going to be the lagging strand, which will have the Okazaki fragments with many primers, but we're just looking, and then they'll all be glued together, but eventually you're going to have these primers at the very ends of the molecules, and that's what we're looking at, because remember it says that the usual replication machinery provides no way to complete the 5' ends, so repeated rounds of replication produce shorter DNA molecules. So let's look at this. We replace DNA polymerase 1, we'll replace the primers, so they'll take off the primers. But what happens, if we look next here, is, let me redraw this here. So this is this one. So this is your 5', and this is your 3' end. And this is your 3', and this is your 5' end of this molecule. So you have your DNA replicated, but when we take off the primer, DNA polymerase cannot add to the end. It has to have a pre-existing. So you took off the primer here, but we can't replace it with anything. So we have a missing piece there. Then when we look at this side, all right, so this would be, it's kind of hard to, it's kind of going in 3', this is the 5'. And so again, you have your 5' end here, and your 3' end here. I'm going to replace and take off that primer, so I'm going to leave a little spot here to show that that primer is missing here, and we can't add it. The DNA polymerase can't add that. So that's what it means by shortening ends. And so then the next time this replicates, it'll be a little bit shorter and a little bit shorter. Every time the DNA replicates, the end gets shorter, the 5' end. And so a protection measurement is at the very end. We have these things called telomeres that are non-coding pieces. So what happens is if you have these TTAGGs all making up the DNA at the end, and the genes don't start till like right here, you have some room where, like if this is your first gene to make a protein, you have some room where it keeps on getting shorter, where it's not going to interfere with making a protein. But once the replicates enough where the DNA replicates and keeps getting shorter every time, eventually it's going to start eroding those genes and make a difference in what proteins you make and so on. And so that happens the same thing over here. So if the first gene is right here, it has these telomeres that adds that buffer in there. All right, so that's the beauty of the TTAGG, this nonsensical kind of order of bases that don't code for making a protein at all. So if chromosomes of germ cells, these guys are, germ cells are cells that divide to make egg and sperm cells, become shorter in every cell cycle, essentially genes would eventually be missing from the gametes they produce. And that's a problem. So as an adult, if you're making egg and sperm cells and you keep on doing DNA replication, and your DNA keeps getting shorter with every replication every time, then you could be passing on chromosomes or pieces of DNA via your egg or sperm cells that the ends have eroded, and if they've eroded enough where it's interfering with the genes, then that's a problem. So an enzyme called telomerase catalyze the lengthening of telomeres in germ cells only. And so these are not in other cells. And so these guys here, these guys divide by meiosis to produce gametes. That's what germ cells are. I used it a few times here, so I want to just define it. And where are these? They're in ovaries in females and testes in males. And so we have a protection mechanism completely built in. All right, and so that is the end of chapter 16. You can be working on your review.